Williams Symposium. We have 4 wonderful students, former students, alumni here. And we have a nice variety of times out from Williams and different career types. They'll all have slightly different formats because, for example, if you're working in industry, you can't show lots of data outside your company. And if you've been out a long time, you have more data than if you haven't been out a long time. And so we have, because we have that variety, we're going to have slightly different format talks for each person. But each talk is going to be roughly 20 minutes with 10 minutes afterwards for just chit chat and questions back and forth. But after all 4 talks are done, we're going to head up to TBL 211, where there is a nice food spread that Christine has ordered for us. And so a chance to circulate and eat and talk with everybody in a less formal environment up there as well afterwards. So I'm going to do the initial introduction, but after that, I'm going to ask people who were their thesis advisors to introduce the next people as they come along. So we won't have too much bouncing up and down, which gives short shrift to our first speaker, who will not be introduced by her thesis advisor. Apologies. So ToriSaltz worked with Siobhan Robinson on the effects of stress on opioids and behavior with opioids. And she has since gone on to Columbia University, where she's in the PhD program there. And Tori's going to tell us about what she's been doing.
Thank you. So my name is ToriSaltz, and I'm a PhD student at Columbia University in the Biological Sciences program. And I'm going to tell you a bit about my work on the evolution of behavioral brain states, and also the path that I took from Williams that made me want to study this. Also, feel free to interrupt with questions at any point. So I was Williams class of 2022, and just a little bit about my time here at Williams. I was a psych major with a neuroscience concentration. I ran cross country and track, and I was on the psychology department student liaison committee. I was a member of Lit Review and also a research assistant in the lab of Amy Hain for a little bit. And in the spring of my sophomore year, I joined the lab of Professor Siobhan Robinson. And when I joined this lab, I really felt like I finally found my place on campus. I just loved being in a wet lab, and I was endlessly fascinated with questions about behavior and thinking about what drives behavior for the first time. I remember slicing brains for the first time on the cryostat and just looking at regions that once mediated behaviors associated with reward seeking and stress response that I'd just measured in live animals. And I just thought this was like the coolest thing ever. So these are some photos from my thesis, where I looked at the effects of opioids on stress response. So some stereotactic surgery on the left, and some immunostains, my first viral injection, and then some brain slicing when I injected food coloring into a mouse brain to troubleshoot where my injection was going. And so this is just to show that I really got to explore a lot of new techniques and learn how these techniques could be used to answer questions in the lab. Then after Williams, I went on to do full-time research in the lab of Dr. Ismail Abdusabor at Columbia Zuckerman Institute, studying touch and pain and the wide range of behavior associated with touch and pain. And here I learned a lot of new techniques and was exposed to a huge breadth of neuroscience research. I learned optogenetics and chemogenetic techniques, both ways to control a population of neurons. And I use these techniques to identify the role of the subpopulation of touch neurons in the skin, and also mapped out brain regions that became active after the stimulation of these touch neurons in the skin. I also looked at molecular changes that drove pain-related behaviors after nerve injury in a mouse model of phantom limb pain. So as I mentioned, I was exposed to this huge breadth of neuroscience labs studying behavior at unique angles. And this typically was through the use of a unique model system. So a lot of the science that I knew came from discoveries in mice, flies, zebrafish, C. elegans, like conventional model organisms. But I began to learn more about the use of novel organisms and how these can be harnessed to look at specific behaviors or specific neural processes that animal could provide unique insight into. So here are a few examples of organisms that are studied at Columbia. We have the xenopus, the African vocalizing bullfrog, often used to study social communication. The naked mole rat, which has all this skin with sensory bristles, used to study touch and pain. The mosquito and the rove beetle, which I got to work with a little bit, so I'll talk a little more about that. Then we have the hydra, which has no centralized nervous system, used to study how peptides can initiate nerve nets or cohorts of synchronized neurons that can elicit simple behaviors. And then the elephant nose fish, used to study electrical sensing in animals. And all of these unique nervous systems are studied at Columbia. And so I found this really interesting breadth of unique neuroscience incredibly exciting. And this motivated me to do a PhD at Columbia Biological Sciences to pursue this interest in animal behavior. So in your first year of grad school, in many programs, you get to rotate through a few labs before deciding where you want to do a thesis. And I wanted to explore this wide breadth of nervous systems in vertebrates and invertebrates to study behavior from unique angles. So in mosquitoes, I studied feeding behavior. Mosquitoes feed on blood and sugar, but process these sources differently, and the mechanisms that drive blood feeding versus sugar feeding are also different. So I studied the molecular drivers of different types of feeding in mosquitoes. I also rotated in a lab that was just starting to use rove beetles as a model organism. And rove beetles have co-evolved with ants for hundreds of millions of years, have therefore evolved these super interesting defensive behaviors. So I studied defensive behaviors in a chemically defensive rove beetle that can actually eject toxins that are stored in its abdomen in order to ward off aggressive ants. Then I ultimately joined the lab of Maria Tosquez to study innate behavior in amphibians in an evolutionary context. And I'm currently working with the salamander, shown here, for its simplified brain structure, small brain size, and homologous regions of interest with the mammalian brain. So in my lab, we use the salamander brain as a way to get insight into how brains have evolved by comparing cell types and circuits with other species. So the amphibian brain can tell us a lot about the evolution of neural circuits in mammals and birds and reptiles, known as amniotes, since amphibians diverged from these amniotes over 350 million years ago. So if we see a circuit or a cell type is conserved in the amphibian brain and also mammals, we can infer its presence in this guy, the tetrapod ancestor. And cell types and circuits that have been preserved and selected for over these hundreds of millions of years of evolution are clearly very important for life. So by tracing circuits through evolution, we kind of get at that question of what are the necessary building blocks of a brain needed to sustain life and mediate complex behavior that vertebrates need to survive. We can also learn about what building blocks or circuits are needed for life on land when we compare neural circuits in the amphibian to the fish brain versus other land vertebrates, since amphibians are the earliest tetrapods to have made that water to land transition and adapt to terrestrial life. So the salamander is particularly advantageous for studying innate behaviors like feeding, mating, prey responses, and this is because the brain is simplified. So many complex structures in the mammalian brain that have nuclei or layers don't have this in the salamander brain. The salamander brain is also about 1/50th of the volume of the mouse brain, so we can image a lot more of it. We can image really distant regions. And their behaviors are pretty simple and pretty stereotyped. So in my work, I'm interested in the evolution of behavioral brain states. And behavioral brain states can be defined pretty broadly as intrinsic states like arousal, wakefulness, sleep, that are typically modulated in the brain by global networks. So basically the entire brain is involved in an underlying network that determines other cognitive processes like how sensory stimuli are processed. So you, for example, like you might be more perceptive to sensory cues if you're in a state of arousal than if you're like asleep. So behavioral states really allow for this flexibility in behavior that allows us to make decisions based on our internal state and meet our needs. So I look at hunting behavior, and particularly I look at this behavioral switch that occurs when salamanders go from a resting state to an active state of hunting. And this behavioral switch will occur immediately when an animal consumes live prey, like a worm or an insect. So once an animal consumes live prey, they immediately begin this transition from a resting state to a prolonged, lasting hunting state. which is characterized by this kind of like prowling and snapping behavior. So I've been characterizing what happens in the brain during these two behavioral states, and particularly what mediates this switch from passive to active. And just to show you what hunting looks like in these guys, because it's kind of fun looking, salamanders really don't move very much. They're pretty inactive, they're cold-blooded creatures, but when they enter a state of hunting, they have this very distinctive prowling, searching behavior that can persist for up to hours. So for my project, I'm using this really drastic behavioral transition to study the evolution of neural circuits that mediate behavioral state transitions. So my goal is to determine brain regions, circuits, and cell types that are mediating this switch between resting and active states. Sorry. Yeah. And then I can ask, are these neural mechanisms conserved? Are they species specific? And again, when we see a particular circuit or a motif in the brain, a cell type, a circuit, a brain region that's been conserved throughout hundreds of millions of years of evolution, we can tell a lot about the importance of this motif in allowing for complex behavior. So in order to ask these questions, I'm using some techniques to look at what's happening in the brain during this behavioral state transition. And I'm just going to show a few of those techniques. These are very ongoing, not completed experiments that I've done just to illustrate some various ways that we can see what's going on within the brain during this behavioral transition from resting to active. So the first is a CFOS experiment. CFOS is a gene that gets transcribed when a neuron fires, and we can therefore use it as a marker for neural activity. So wherever you see a CFOS positive neuron in the brain, we can assume that that cell was active during whatever behavior was performed before the animal was euthanized. So for me, since I wanted to look at this behavioral switch, I looked at peak CFOS activity at the time of live prey consumption and at various time points after live prey consumption. So this is going to be what a whole brain looks like with CFOS expression. And I quantified CFOS expression in the whole brain after clearing the brain and imaging with a light sheet microscope. So here you can see the entire 3D brain because the sample has been made transparent and is imaged with a thin laser that scans each plane individually to build an entire three-dimensional sample. And then I've used a neural network, an AI model, to detect and count positive CFOS cells in the brain. So wherever you see a little colored dot, that was a neuron that was active at the time of live prey consumption. Then I've made these heat maps, which are CFOS expression in the whole brain, just collapsed across the z-axis. So basically taking that 3D image and just smushing it across the z-axis so that we can visualize the entire forebrain and midbrain. So this is a salamander brain shown here, the forebrain and the midbrain. And then these heat maps show global activity. Red areas are areas in which activity was increased in the brain compared with a control animal that was just resting, unfed, versus blue areas are areas where activity was decreased in the brain. And this is just a visual representation that allows us to see globally what's going on in the brain, what regions are activated, what regions are inhibited, and this will allow us to gain some insight into what is going on in the brain when the animal enters this hunting state. Then another way that I'm able to look at this behavioral switch between resting and hunting is by looking at in vivo neural activity during real-time behavior. And I can do this with calcium imaging. So calcium imaging is a technique that allows us to use fluorescence to see real-time neuronal activity. So when a neuron fires, calcium flows into the cell, and we use these genetically encoded calcium indicators in the cell, which bind to calcium and then fluoresce, and we can detect that fluorescence with a microscope. So here I'm imaging the entire surface of the brain, looking at these really distant regions across the whole brain. And this is typically done in a mouse brain, which is a lot bigger. And also since the mouse brain is a lot bigger, it only reaches this area called the neocortex, which is kind of this large scaling flat region that goes over the entire surface of the mouse brain, and it's very thick. But in these guys, in these salamanders, they don't have a neocortex. So we can look at the entire forebrain and the entire midbrain because their brains are so tiny and kind of stretched out as opposed to deeper and more compact. So this is a head-fixed animal with a cranial window that allows me to see the whole brain here and watch little flashes of activity all over the brain while the animal is in a resting state, and then while it eats live prey and then goes into this hunting state. And so when animals feed live prey, or are fed live prey, I see this drastic reorganization of the forebrain, meaning rhythmicity and regions of activity are completely altered. So before feeding, we see this continuous firing of this region called the medial pallium, which we think is mediating a resting state by kind of tonically inhibiting locomotor regions. So the medial pallium projects to a region called the basal ganglia, which is involved in locomotor control. And we think that these really stark waves followed by a period of silence are inhibiting locomotor motion of the animal during resting state. Then in the feeding, after feeding during this hunting state, we see a lot of asymmetry of the two hemispheres. We see decreased medial paleo waves and asynchrony in regions. But essentially just a complete transformation of the forebrain, which is exactly what we'd expect for a behavioral brain state. We'd expect that there are these global networks that involve distant regions, underlying states like resting, that are changed when the animal becomes more aroused, and that these global networks may modulate how sensory information is processed in the brain and how decisions are made. I'm just going to scroll through the rest of this so you can see the stark difference between hunting and resting.
Or how is hunting happening in the Hedwig's?
So we're counting on the fact that live prey induces a state in which the animal is more like hyper aware of sensory stimuli, and may kind of have this brain state regardless of whether it's actually freely behaving. But when we image the animal, we put it on the microscope, and then take it off, allow it to hunt, record behavior, and then put it back on for periods of time. Yeah.
Is there any visual stimulus, like while it's being undergoing calcium imaging, like is it seeing prey?
So we can measure the point at which the animal is given a piece, like a live worm in front of it, and then afterwards. But there's a lot of confounds at which the animal is actually consuming live prey, because that could be like food consumption related areas. So I've been trying to not take into account like a 10-minute chunk of hunting behavior and just kind of let the animals consume live prey, go into a separate tank, hunt in a tank with no sensory stimuli, and then hope that this kind of intrinsic network appears to still be propagating some type of motor behavior without the presence of any sensory stimuli, which would be more of like a behavioral state as opposed to like food keeps appearing and the animal keeps tracking food.
Yes.
Yeah. So I just showed two ways that we can look at activity in the brain during this behavioral state transition from resting to active behavior. And the big question is still, how did these brain states emerge throughout evolution? And so by uncovering brain regions, circuits, and cell types that are mediating behavioral state transitions in the amphibian, we can compare these to what we know about other vertebrate classes and determine brain motifs that have been functionally conserved over hundreds of millions of years to generate flexible, behaviorally flexible, complex behaviors. And then we can get at that big question of what do we need to build a brain, or how did evolution build a brain that is capable of flexible and complex behavior? So with that, I want to thank you all for listening in my lab. And I'm happy to answer any questions about my research or just grad school in general.
I was doing research here, and obviously a big motivator to continue doing research afterwards, but did you have any like summer experiences, a post-bac, or like other experiences away from Williams that really solidified your desire to do a PhD? And like, are you from New York? Why was
Yeah.
That like your choice?
Yeah, so I am from New York, and that was a big motivation to stay in New York. And I did do some summer research that kind of exposed me to like a big institution. And I thought that was really helpful in solidifying wanting to pursue research. But I also think that doing research here is really impactful because, you know, like when you're an undergrad at a big institution, you're kind of working for a grad student or postdoc. And here you're often like generating ideas and you get a lot of freedom to explore. And that's much more what it's like to be a grad student. So I actually think that the small college research experience was much more impactful for me.
I have an obnoxious research question. I was really struck by the asymmetry
Yeah,
that came in when they started the hunting behavior.
yeah.
Do you have any ideas about why it
So
should go from symmetrical to asymmetrical?
yeah, so I think it's a big question about why this also happens in the mammalian brain when mice become more aroused when they go from a resting state to a higher arousal threshold. There's also much asymmetry between the two hemispheres. And a lot of these like kind of whole brain networks that are mediating arousal were often written off as like noise in this type of imaging. So I think it's a kind of new thing that people have the tools to look at these networks and dissect them on such a large scale. But it's a common theme throughout multiple vertebrate classes that the brain becomes more asynchronized and more asymmetric as an animal becomes more aroused. But I don't think
Yeah,
that
I was just
we
thinking
understand
about all the
why.
research that says that some species actually sleep half their brain at the time. So the.
Yeah, there is an interesting like interhemispheric competition that occurs during sleep where the midbrain sends projections to areas of the forebrain and one area will undergo REM sleep before the other area. And I think sleep is like super pertinent to this type of work because like we don't really know how amphibians sleep. There may be like some kind of like clustering of like little bouts of sleep, but maybe not just like a chunk of sleep the way we do. So it's very possible that this resting state is much closer to a sleep state than just like a resting state, and that maybe this arousal state is more like an awake state for them.
Yeah, I was just thinking migrating birds and long distance swimming marine mammals turn off one hemisphere at a time
Interesting.
while they're actually moving.
While they're moving. I guess if you're migrating for so long, there has to be some kind of period of rest.
You sleep one side,
Yeah.
and you sleep the other side.
Yeah,
So after Williams, you did a year of post-bac research.
yes.
Can you think back to your senior year? And how many post-bac jobs did you apply for?
Yeah.
What made you apply to those? How did it feel doing that senior year, that kind of experience?
Yeah, so, well, you and Siobhan Robinson helped me a lot in applying to postdoc positions. One of the hardest things was just like, how do you phrase an email? And you sent me an email that I now have, and I'm happy to share with anyone on just how do you like contact, how do you make first contact? It's kind of scary when you want to do research, and how do you express interest in someone's research that makes them want to write back to you. So I think I sent out 5 or so emails, and it definitely helps if you have like a reason for being interested in that lab, like if they can sense that that kind of fits with your interests for a while. And then I had a couple of interviews and decided that the lab of Ismail Abdi Sabor at Columbia would be a really good fit for me and also help me pursue my later goals of doing a PhD.
Was that like a completely cold email? You hadn't reached out at all, or you weren't actively applying to a program? You were just like, I would like to work with someone, and you
That was actually a cold email, yes. But I did have other circumstances where a professor connected me or someone else introduced me. Yeah. And you can also be introduced by not a professor if you happen to know a student or a postdoc. It sometimes helps to just even have someone say like, hey, someone from this college is going to email you, look out for that, because PIs are very busy, they get a lot of emails, so little things like that can help for sure. Yeah.
Can you talk a little bit about like what your schedule is like in grad school?
Yeah.
And like, the hours?
Yeah, so my program is mostly research, not class focused. So I'm in the second year of my program. The first year of my program, I was rotating through different labs. So I really started in this lab July of this past school year. now that I'm mostly done with my classes, I'll have one class this semester. And most of my days will just kind of look like going into the lab, working a full day and, yeah, that's pretty much, it's not that different from, a regular job, except that I think there's a little bit more, like, when some people get to like kind of feel like they can log off for the weekends, sometimes there's a little bit more of like, well, like, that's just gonna harm you later. So I think there's more, personal investment in what you're doing than like a typical job, but sometimes that's really nice.
Thank you very much.
Thank you.
I'm gonna introduce the second speaker, who also was from my lab. I'm sorry, Tori, that I didn't do your intro. But I'm so happy to have both of them here, and I'm so